I'm on the hunt for one of Australia's most venomous animals. Its sting can cause unimaginable pain and sometimes even kill. But it's because of its deadly nature that scientists want to get their hands on it.
There's one, there's one, there's one. The crowd goes wild.
Oh my goodness. Because hidden inside venom could be a medicine of the future.
The things that they're doing are just spectacular.
So, I'm off on an adventure to join researchers as they seek out nature's most dangerous creatures. I'll see how they're harnessing venom.
My heart is beating quite fast and I'm not even involved.
So that it doesn't kill, but cure.
We've shown that it actually prevents further brain damage. It's very exciting, yes.
And ultimately, it'll take me somewhere I never thought it had a place.
And is this the heart?
This is the heart.
From something that evolved to inflict pain, paralysis, even psychosis and death, could come revolutionary new medical treatments.
This is the story of
Venom.
I'm on a quest to uncover the secrets of venom. To find out why scientists believe it holds the key to a whole new pharmacy of drugs.
I'm feeling excited but cautious about this adventure because there's an innate fear in me about venomous creatures, and knowing that I'm going to have to be up close with them, you know, it gives me a little burst of adrenaline. So I'm feeling really happy that I'm going to find out more, but also a little bit anxious.
I'm getting off this ferry at Gari, or Fraser Island. In the language of the traditional owners of this land, the Butchulla people, Gari means paradise.
Galango Jali, Galango Gari, Wanyanyanyanyangu, Wanajinanga.
Lying 250 kilometres north of Brisbane, this is the largest sand island in the world. It's a paradise, not only for tourists, but for all sorts of venomous creatures. So it's the perfect place to start my journey. I've come here to meet a group of venom researchers from the University of Queensland.
Because it's a sand island, it's a completely different ecosystem. So for funnel webs, it's much easier to dig them out of sand than it is to dig them out of clay.
They call themselves the Drugs and Bugs team.
We had to have some really interesting molecules that we think might be therapeutically interesting.
This island is amazing. It's full of things that can envenomate you with big fangs and pincers and all sorts of apparatus. And the Bugs and Drugs team are keen to find more and more venomous creatures because they've found that the venom they've collected so far could be revolutionary for our medical system.
And the deadlier the venom, the better, for biochemist Glenn King.
The main goal is to get more funnel web spiders because we don't have any, but we're looking more broadly for other venomous animals that we've not collected before, like assassin bugs, centipedes, scorpions, wasps, ants.
So everything?
Pretty much everything.
Everything that's people's worst nightmares, generally. That's...
Some more so than others.
Glenn's team can hijack the chemicals in dangerous venoms and redirect them towards saving human lives.
We're interested in trying to develop drugs that target the nervous system. And if you think about the way that most invertebrate venomous animals work, is they're trying to paralyse their prey. So over hundreds of millions of years, they've developed these neurotoxins that target the nervous system of their prey. But those same molecules that they use to capture prey can actually be useful in the context of human therapeutics.
So what should I be looking for as I move across?
You got to look on the ground, you got to look in the air, because there's a beautiful golden orb weaver there, right? And so that's the thing that people often get scared of, because they run into them in the night, in the dark.
Yeah.
But they're actually beautiful spiders and completely harmless.
But not a venomous creature.
Oh, absolutely venomous, yeah.
Oh, it is venomous?
It is venomous, absolutely venomous. No, quite venomous. Not to us, but it's quite venomous. Catches the prey by envenomating them with neurotoxins. And we think the web actually might be venomous as well.
No way.
There are venomous creatures at every turn on Gari, and Andy Walker has found one bug with something highly unusual in its armoury.
So today we're looking for some assassin bugs. And they're tiny venomous creatures, but we don't know very much about their venom.
But they're called assassin bugs.
Most of them are ambush predators.
So what sort of tree in particular are the assassin bugs fond
Well,
of?
we were finding a lot of them on these little tea trees, like we can see over here.
All right, so tea tree, big brown buggy thing. Okay, this is much harder than I expected. I think I've got him or her.
Oh, you found something?
I think so. It's very still.
Oh, yeah. Yeah, beautiful.
So it's an absolutely stunning little creature. It's got these beautiful sort of angles on its abdomen at the back, but the head is crazy because it almost looks like it's got an elephant trunk tucked in.
Yeah, that's right. So those are these piercing, sucking mouth parts. Assassin bugs use them firstly to inject venom and also to suck up the juices of animals through this tiny straw. So one of the jobs of the venom is it has to liquefy the insides of the prey into this kind of soup it'll suck up and eat. And also, if you were to really harass her, she would use it defensively to give you a really nasty sting as well.
So the venom in this case has many different jobs to do. Does that equal more complexity?
Yeah, we think it probably does. The other thing we found out about this is something really unusual, which is that they actually have two different venom glands inside their thorax and abdomen, and they've got this complex network of different valves and tubes that allows them to inject the contents of either of those glands as they see fit. And some of those we think to do with paralysing prey. Some are probably defensive, and some have the job of liquefying.
So am I understanding this correctly? It's got two different venom glands. They have different venom in it, and it can sort of decide what it wants to inject.
Absolutely, yep.
That's incredible.
But there's another creature on this island with a far more complex venom. A toxic cocktail that has evolved over 400 million years.
If you grew up in Australia, funnel web spiders were probably the reason that your parents told you not to go digging around in the garden.
But today, those spiders are the reason that we're going to dig in someone's backyard.
You sometimes see one under stuff like this, don't you?
Because they're thought to have one of the most potent venoms in the world.
There's one.
Look at this.
Yep.
It's a good burrow. Look at that.
No
Quite
way.
big too. It's a very big one actually. That's an unusually large hole.
And I like that you brought so many people to dig because it means that I don't have to. But who's going to do the honours today?
I think David's going to do the honours.
All right, well, I'm going to give you some space in case it turns into like a Tasmanian devil type situation.
All right, David, take it away. Absolutely.
Having dug up more than 70 funnel web spiders, David Wilson has a clever way of finding his prey. Tracking the spider's delicate web.
It'll be sitting in a little sock at the bottom of the burrow, and what David's going to try and do is follow along the edge of that funnel.
So there's another one right over here.
Wow, another one already?
Yes, there's
That was
another
quick.
one right here.
Oh, well done.
It's like about 5 metres away.
The find is so soon and so close. I'm starting to feel surrounded.
How did you manage to find that?
Well, you can usually spot the funnel webs fairly easily because they've got these distinctive webs, often at the base of a tree, with these trip lines. And the trip lines signal to the spider if prey is walking by. So the spider will live in the burrow and then come out and grab any insects that happen to walk close to the burrow.
Right, grab it and pull it back in the burrow.
So did you just let that spider know that we're here?
Yes.
It was like a door knock.
Yes.
Hello.
Knock, knock. I'm coming. Coming for you.
Give us a shout when you get close. It's unnerving watching David getting closer and closer to a creature with a venom that could potentially kill him. Can you tell me the main effects of a funnel web venom? What would happen to me?
So what it does, it's very unusual venom in that it sends electrical signals across your entire nervous system, so everything turns on. So you sweat, you salivate, your muscles fasciculate. Eventually you get system collapse and you die typically of respiratory failure if it gets that far.
Ooh, I'm seeing some large-scale action. Did you have an inkling that it was going to be quite that deep?
No. Because it's still going.
It's still going. The hard work is worth it because from a human therapeutics point of view, funnel web spiders are a molecular goldmine. So funnel web venom, in terms of the broad gamut of venoms that there are in the world, how complex is it?
Yeah, it's one of the most complex that has ever been seen. Up to 3,000 molecules in the venom of a single spider. What's remarkable though is that only one of those molecules is responsible for the human envenomation syndrome, the thing that kills us. It's just one molecule of those 3,000. So if you took that molecule out, that venom would be actually completely harmless. And that's why we think they're still worth looking at from the point of view of human therapeutics, because there are many molecules in there, or most of the molecules in there, are harmless to humans and might have some benefit.
That's a lot of process of elimination though, isn't it?
It is. In our collection we have venoms from over 700 different species, and then once we find a venom that has the activity we want, the trick is then finding the molecule within that venom that is giving you that activity.
So in a sense this digging and finding the venomous creatures is only a tiny part of the work.
Just at the very beginning of the project.
Got it.
You got it? Something there. Oh, it's actually not that big. Well done, David. Fantastic.
I thought you were going to put your hand right down there. Oh my goodness.
No, don't do that, David, please. Pull it out with that very technical instrument called a spaghetti spoon.
What? Hello.
Fit the torch on.
It's not overly big.
Yeah. Wow.
Which is a little bit surprising given the size of the hole and how far down it was.
Yeah.
Juvenile.
Wow. It's possibly never been outside in the daytime.
No. It's probably freaking out a little bit at the moment.
Yeah. Does that mean that it becomes less predictable, sort of, or?
Yeah, they become a little bit erratic. So they may just freeze or they may scatter around a bit and then suddenly go into aggressive pose.
Lucky they're not known as the fastest.
Well, I think it's time to put this in a tube.
It's so cool, like stealth, black, shiny, and a little bit angry. Well, congratulations.
Yeah, well done. Well persevered, David.
The spaghetti spoon won again.
How are you guys going?
No luck here.
No luck?
Oh, okay. Do I have to hold the...
You can hold the prize.
It seems like a lot of effort for the promise of just a few tiny drops of venom. But within each droplet, there's a vast cocktail of toxins, and each ingredient has potential medical benefits. Gari is an amazing place, and I've learnt so much in these few short hours that I've spent on her shores. Venom is so much more complicated than I ever expected. So much more that even the scientists are struggling to understand its complexities. And they want to find out more, and in turn, they've infected me. I want to find out more now. I'll explore the medical potential of funnel web venom a little bit later. But to understand what it is about venom that has scientists so excited, I need to go back a few steps to explore why it evolved to become such a deadly substance to prey, and sometimes to us. Australia is infamous for having more deadly snake species than any other country. While they stay mostly hidden hunting for their dinner, they do occasionally cross paths with people. And the consequence of being bitten can be devastating.
Hello? Knock, knock.
Hello.
Hi, I'm Anne.
How are you? I'm Hamish. Nice to meet you.
Nice to meet you. Are you the snakebite guy?
I'm that infamous guy, yes.
Zoologist Hamish Tristram is an experienced snake handler.
This is my snake room in here.
So, when he was bitten by his red-bellied black snake Jet, it was a bit of a shock.
Jet, as you can see, is down here.
Oh, I can see
This
his
is my
tail.
one of my favourite snakes, yes.
So tell me, what happened?
Well, look, this snake here I've had for quite a number of years, and there was a day basically where he mistook my finger as a food item. Basically, he'd tipped his water over, and so all the paper was wet, and I thought, I'm in a bit of a rush, but I'll quickly change that water. So as soon as I opened that door, he thought there's a rat coming, and bang, was onto my finger. Now, at that point, he gave the finger a good chew and injected a fair yield of venom.
And that venom did some serious harm.
It literally felt like a hot poker going through my finger. I could feel that pain, and then I put the bandage on. I woozily stood up and I made my way out into the living room here. And in fact, initially I collapsed because I was just so faint. I was feeling really sick, I was vomiting. Even my bowel control was starting to go because my body was literally trying to get all these toxins out of my system.
The symptoms Hamish experienced were due to the black snake's chemical weaponry, designed to immobilise quickly with devastating effects.
All species of snakes have different properties in their venom according to the prey items that they naturally prey upon and the habitat that they live in. So for black snakes, they have what we call a low neurotoxin, but they have a high myotoxin, which is a tissue and muscle destroying component, very effective against frogs, which is their primary prey item. So that venom there is one that causes that tissue destruction, that necrosis, and also to pre-digest that prey item, to break that tissue down to ensure that digestion is easier. So unfortunately, it broke down my finger as it was meant to the prey item. Had this have been the rat, that's the exact result the snake wanted.
It is an absolutely beautiful example of how the venom is meant to work. It's meant to disable a prey item.
Look, that's exactly right. And the thing is, it is a fascinating subject. I mean, I think in Australia, look, we're known for our venomous snakes and the toxicity of our snakes, and people tend to shun them, and we should really embrace it because they are an amazing example of evolution and how that animal has become that for a very specific purpose.
It was a lucky escape for Hamish. A different venom could have had a vastly different effect on his body. How venom has evolved to target specific prey is something that snake lover and molecular biologist Brian Fry has been exploring.
How's it going, laddie? What in here? Water, first aid kit in the back, headlamp, torch, shock blanket, pretty much anything we might need.
We've come to a snake hotspot in Queensland with reptile wrangler Lee Davis, so I can learn how snakes like the eastern brown have become one of the deadliest in the world.
So one of the tasks of a local snake catcher is actually releasing snakes, because once you catch one, you've got to release it. And one of these brown snakes that's in this bag was actually caught up in a net and got a little cut. We've given it some antibiotics and made sure it's all healthy. And now we're getting ready to release it, which is a pretty lovely thing to see. So what do we got? 2 browns, biggies or littles? Oh, hello.
Oh, aren't you a darling little
That's
thing?
such a pretty one. So what's the story behind this one then?
He was caught in cast netting. You can just see a bit of roughing around
Oh yeah,
The
about 20%
scalation
down.
just about there.
That kind of netting can cause really severe injury to
Really.
snakes.
If the people hadn't rung about the snake stuck in the cast net, it would have definitely died there.
Brian wants to see snakes thrive, as they're a vital part of the food chain.
Gee, the colour is stunning.
They're also vital to our well-being.
This is what frustrates me when people say, oh, the only good snake is a dead snake. Well, no, because first off, do you like having a lot of mice around your place? And if the answer is no, then you want snakes. But also as a source of life-saving medications, where if you know of anybody, for example, who takes high blood pressure medication, odds are they're taking captopril or one of its derivatives. Well, that's a medicine that was developed 40 years ago from a modified snake toxin. It has been one of the top 20 drugs of all time. Its medical importance cannot be overstated. It is up there with aspirin. And if we lose magnificent snakes like this, what other wonder drugs could we have lost?
The eastern brown has one of the most potent venoms on the planet.
They're the only type of snake that has regularly killed people in 15 minutes or less. A brown snake this size is going to absolutely nuke your blood.
I think it's angry.
That's an incredibly quick death, considering we're not their primary prey.
So, well, from an evolutionary point of view then, why have such potent venom?
It's a very dangerous game that they're playing, particularly animals like brown snakes and taipans that are feeding on mice and other rodents, as well as things like bandicoots, which are comparatively very dangerous prey animals relative to snakes that are feeding on like frogs or geckos.
Right, because they've got teeth and claws.
Exactly. So they need to overwhelm their prey. So the speed of action becomes paramount.
And so they're not after us, it's just sort of a side effect of it that this is so toxic to humans.
Exactly. So the most dangerous snakes to humans are the ones like adult brown snakes or taipans of any age that are taking mammals as their specialist prey animal, because their venom is now honed to a prey animal that's much more evolutionarily closer to us than an animal that might be feeding on lizards.
It's always nice to release them.
To find out what happens if we're bitten by a snake that's evolved to hunt mammals, Brian is taking me on a trip to collect venom from a coastal taipan.
I can handle them in the hook for a little bit. And go.
Yeah, nah.
The coastal taipan is the most dangerous species of snake in Australia.
How long is he?
2.6 meters.
Oh my goodness. I can see his muscles flexing.
Until the development of an anti-venom, its bite meant certain death.
And that's not blood on the ground, by the way. That's paint from the previous homeowners.
But that doesn't seem to concern medical researcher Christina Zdenek,
So this is Squishy.
Hello, Squishy.
who's about to collect its venom.
My heart is beating quite fast and I'm not even involved.
So sometimes he's reluctant to bite.
So you just encourage him to bite down on that?
Yeah, as soon as we get his fangs in there.
Oh my God. Wow. Good on you, Squish.
So he has about 12 to 14 millimeter long fangs.
That was a huge amount of liquid came out.
Yeah.
And it's got compressor muscles on the side too, so that press it up against the bone. So it has a syringe-like action, is propelled under incredible force, over 100 lethal doses on any given milking.
So, Christina, where is all of that liquid actually made in?
So it's got venom glands just behind the eye here, and it's a specialized gland. So there'll be a myriad of toxins in there, up to 100, 150 different types of toxins within a venom gland, and of course designed to immobilize prey.
To see what happens when those toxins get into the bloodstream, I'm going to take part in an experiment.
They leave a pretty looking corpse.
One where my blood is going to be envenomated.
So I suppose it's time that I made a sacrifice for science.
Thankfully, the only puncture wound I'm getting is from this needle.
the venom.
So I'm going to show what coastal taipan venom does to the blood. Now, of course, this is a smaller volume than what's in your body. So this will actually simulate what happens to prey items as opposed to inside a human body. So the first step is I'm going to pour one of these vials into one of these jars.
This is liquid gold here.
Christina pours my blood into 2 jars. One will be mixed with venom and the other water as a control.
So that's going to prove that it's not just the fact that we've taken it out of your body.
And then it's toxin time.
So here is Squishy's venom that we extracted recently.
And in less than 3 minutes...
So as you can see as I go to pour, so we can demonstrate what's actually going on.
Oh my God, it's really solid.
That one is turned into a piece of jelly, whereas the control is still liquid. There we go.
Fascinating, that was so quick.
Here's where the action of venom gets intriguing. While small amounts of blood, like in a rat, completely coagulate, it works differently in the human body.
It's a bit opposite, and it all comes down to the volume of blood. Now, inside you have liters of blood. So the venom is very dilute throughout your entire body. So it does create small little clots, but it's not enough to cause a stroke. Instead of creating one big clot and stroking out like a prey item would, you'd actually bleed out internally.
And it's this effect, the ability to clot blood but not block blood vessels, that has scientists keenly interested.
So is there a potential, medical application for that though? Because clotting, making more, circulation, these are things that we all need in our daily life.
Absolutely, and there's already been a case where there's a very procoagulant venom like taipan, but it's actually from the eastern brown snake, and there's a protein, a toxin in their venom that's currently being patented, and it's in order to stop people from bleeding out during surgery. So doctors can actually use the venom at the site where they're trying to prevent bleeding from occurring. So it absolutely has great potential medical uses.
That's so cool.
And because venoms from different animals affect different systems in the body, there's a whole range of toxins that could be useful in medicine.
Just about everything you can think of. So depending on the species, some of those effects are myotoxicity, so affecting your muscles, bursting those cells. You've got neurotoxicity, preventing your nerves from acting as they should normally. Coagulotoxicity, as we're seeing with this coastal taipan. We've also got cytotoxicity, affecting your cells. You've got hemotoxicity as well, affecting red blood cells, and blood pressure, and also cardiotoxic toxins as well that affect your heart specifically.
So there's basically not a system inside a body that some sort of venom somewhere doesn't act on.
Absolutely, thanks to evolution.
Thank you, evolution.
Because of its danger to us, snake venom has been well researched over the last century, and it's now known to contain up to 100 different toxins. But there are venomous creatures that remain a mystery, even after decades of research. Some of the world's most dangerous venoms are produced in minute quantities that punch well above their weight. And I'm on the trail of one.
So the thing with this venom, right, is that it's so crazy that it's got a psychological syndrome named after it. It causes incredible pain and an impending sense of doom. Great.
The tiny creature that produces it is named after the Irukandji people of the coastal areas around Cairns in Queensland. I've come here to search for the infamous Irukandji jellyfish.
How are we?
I'm good. I'm Anne. Nice to meet you.
with Jamie Seymour, who's trying to understand how its toxins wreak chemical havoc on our bodies.
Couple of waves coming.
Whee! So tell me and my mother why I'm out here at night.
What we found about 15, 20 years ago was that they're attracted to light. So if we come out here at night and we know where they are, we can put lights in the water, we can then take them home and extract the venom.
Jamie has a very personal history with the effects of Irukandji venom. He's been stung 11 times.
Complete total pain right throughout the body.
Once, when he was out with a film crew.
Oh dear.
So what is it like being stung?
Imagine the worst thing on the planet and you're not even close. For me, I get pins and needles in my joints, as if someone's poking red hot pokers in there. Feels like an elephant sitting on my chest, so you have trouble breathing. You feel nauseous, you're throwing up for 3 or 4 hours. And you've got this pain that just keeps building.
But it could have been much worse. Irukandji stings have been known to cause brain hemorrhage and death.
Overriding the whole thing of that is this feeling of impending doom. This feeling of, this is going to go south and somebody's going to have to kill me. I mean, it's you wouldn't wish it on your worst enemy.
Then why do you keep on researching?
I haven't been stung since 2001, 2002.
So you've forgotten?
Pretty much. Pretty much, yeah.
The tiny Irukandji can be elusive, but for Jamie, that's part of the attraction.
We come out every single night, and I have to tell you, we learn something every single night. Every time you think you've got it under control, you get another piece of data which just goes, yep, Seymour, you haven't got a clue. Okay, so now we're here, we're going to try and catch them, okay? So what we're going to do, basically we've got some underwater lights.
Yep.
We're going to turn them on, drop this over the side of the boat, and then with a bit of luck, the jellyfish will swim past and we'll bag one.
So will they come right up to the light?
Right up to the light.
The lights are barely in the water.
Oh, look at the squid in the water here.
And weird and wonderful creatures begin to emerge from the deep. But so far, no little jellyfish.
Oh, look at those little fellas.
No, do not tell me they're better than jellyfish.
No, they're alien looking,
I'm going to throw you overboard.
Look at the beards.
Okay. Oh, netrostoma. Wow.
Yeah, what is that? It looks like a moon reflection.
Yep, that's a jellyfish.
Really?
Not the one we're after.
No.
It's a thing called netrostoma.
It's like a swimming brain.
It's starting to seem like tonight might not be our night. But then, at last...
There's one. I win. Right down there. Oh, lovely, 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 lovely. Just here. See him?
No.
Oh, just over here. Winner! The crowd goes wild.
No way.
Touchdown. And there you have one.
It's tiny. Oh my goodness.
And he's not fishing at the moment, so his tentacles are only quite small.
Wow.
So if he was fishing, those tentacles would be 3/4 of a metre in length.
I can't even see it when it's on there.
Yeah.
The crazy thing is, it looks like it would be impossible to harm you, but it's probably one of the scariest things that we have in our oceans.
So now's the time where you go, okay, I need to have my brain switched on and think about what's happening. Everything you now need to think about has possibly got jellyfish tentacles on it. Now, you only need 1 or 2 millimetres worth of tentacle contact, and we're seeing you in hospital.
And they're tiny. I mean, so what sort of prey are they actually going for?
Oh, these guys are hunting larval fish.
So what's the delivery system for this venom?
So they have what's referred to as nematocysts. So they are basically cell organelles. So they're part of a cell. Think of it as a wound-up hypodermic needle inside a balloon. So when that comes in contact with you, that hypodermic needle fires off. It's a really interesting system. It's the fastest biological mechanism that we know of for this. When this thing fires off, it fires off quicker than a bullet out of a gun.
And it's not just the delivery system that's impressive. It's also the variety of venoms found in just one organism.
They've got venom in their tentacles. They've also got venom on their bell, the body of the animal, which is a different type of venom. The venom is different for juveniles than it is from adults. The venom varies from geographical locations, and it varies from different times of the year. It's 96% water. The bottom of the evolutionary tree has 24 eyes. How could you not want to study that?
Jamie and his team have discovered that though Irukandji are relatively simple animals, their venom is packed full of toxins.
The thing with these venoms as well is they are quite complex. So if you compare them to snake venoms, and snake venoms are complex, but nothing like these. I mean, certainly we reckon that the work that we've done is somewhere between 150 and 200 different proteins. Just proteins alone in the venom. And we haven't got a clue what any of those do at this stage of the game.
Such a complex creature. I can see why you've spent so many years.
Well, as I said, remember that it's 96% water, but the things that they're doing are just spectacular.
The Irukandji show how versatile venom can be, and that size has very little to do with potency. But it's the dramatic effect the venom has on our bodies that holds the promise of exciting medical breakthroughs.
Certainly the other jellyfish species that we're working on, we've found some really cool things that would be good for drugs, but who knows what's in this? I mean, if for example, we think that this is stimulation of your adrenal system, if we can find the bit of that venom that does that, remembering that venoms are cocktails, so if we can pull out just the little bit that stimulates your adrenal system, we might not have to give you adrenaline anymore. We might be able to give you that to stimulate your adrenal system. I mean, it's endless, the questions that you can ask and the answers that this animal at the bottom of the evolutionary tree has that's been doing this for millions of years. We as chemists have been doing it for a couple of hundred.
When it comes to nature, we can be pretty selfish. We think all about how nature is going to impact on us. But when it comes to venom, we need to get over that. It's really unlikely that you're going to die. We need to rethink how we come at venom. It's this amazing, complex set of molecules, and it could just save your life.
Scientists are getting closer to unlocking the potential of this pharmaceutical frontier. Recent advances in technology are helping them decode the biological structure of even more complex venoms. Back from Gari, the Drugs and Bugs team have set their sights on unlocking the deadly funnel web venom.
So Sam, I know you name your spiders. Does this one have a name?
This is Colossus.
Colossus!
Because she is a truly impressive specimen. So she's actually tucked away in her burrow at the moment.
Oh wow, look at this. I'm fogging up my glasses. I think that means you're making me slightly nervous, Colossus.
All right, so I'm going to transfer her into this container.
Oh. What's the biggest challenge in terms of milking a spider?
So spiders actually don't want to give up their venom if they don't have to. It takes a lot of energy for them to make it. It's really, really chemically complex. And they need it to protect themselves from predators, and they need it to catch their prey.
To get Colossus to release some of that precious cocktail, researcher Samantha Nixon is giving her a little encouragement.
So there she is there, trying to show how big she is.
Well, she is big.
Don't mess with me.
Look at her big fangs.
So you can see she's flexing her fangs.
She can move those fangs, like articulate them.
And she'll eventually actually drip venom from her fangs, and then I can suck it up with the pipette.
She is terrifying.
Oh, there we go.
Oh, wow.
So you see that tiny little drop right on the end of her fang? So it's coming out of both fangs now.
Oh, that one's a real big drop. Wow, a minute amount of liquid. That is incredible.
All of these bubbles are because it's so protein rich.
Thanks, Colossus.
Just a few drops of this venom will keep the research team busy for many months. What they've discovered so far is that funnel web venom is made up of a huge number of active molecules.
How many molecules did you find? How complex was the venom?
Actually, upon a more recent analysis of the venom with more up-to-date technology, we've discovered roughly 3,000 peptides just in this one venom.
Each cup Natalie Sayers is laying out represents a single molecule.
This is a lot of cups.
It is a lot of cups.
And the molecules they're most interested in are proteins called peptides.
Peptides, what are they?
Peptides are just small proteins, mini proteins. They tend to be very specific to their targets.
They're found in every cell and tissue and perform a wide range of essential functions in the body.
So of those 3,000, do you know what they do?
In fact, the majority of these, we don't know what they do. So much unexplored space.
Yeah, it really is a new frontier, isn't it?
For sure, yeah.
Because they control our biological processes, they're ideal candidates for new, targeted drugs. But to find one with potential amongst the 3,000 required some good luck.
Actually, when we were looking at all the different sequences, one of them really stood out to us. It was very similar to a peptide I'd worked on during my PhD, and we had an idea that it could potentially have amazing therapeutic implications.
What's it called?
This one is called HI1A.
HI1A.
Correct.
And it's in here somewhere.
It's in here somewhere.
All right, let's see if I can find HI1A.
The structure of HI1A was similar to a peptide from a South American spider that had previously shown promise as a medicine for brain cells.
I think it's it.
That's the one.
Studies in the laboratory revealed it could be used to reduce the impact of one of the world's biggest causes of death, stroke.
In rat models of stroke, we can give the peptide post-stroke up to 8 hours after a stroke, in fact. And we've shown that it actually prevents further brain damage from occurring.
Right.
So, in theory, we hope that this molecule will be saving brains.
In humans?
In humans.
That must be like a eureka moment?
For sure, yeah. It's very exciting, yes. Very proud to be a part of the research.
Since that incredible discovery, Glenn King's team has been working to develop a revolutionary new drug.
This almost looks like molecule.
Based on the way HI1A works in the body.
And this here is the largest nuclear magnetic resonance spectrometer in the southern hemisphere.
Wow.
With a magnetic field that's about 400,000 times more than the Earth's magnetic field.
My God.
And what we use it for is determining the structures of molecules, including HI1A.
So you've got all of these amazing people and technology working on these molecules like HI1A. What sort of thing could it be used for in a health context?
So what we're trying to develop it for is to treat conditions of ischemia. So this is where a tissue is exposed to low oxygen conditions for some period of time. And the classic examples are a heart that's been taken out ready for heart transplant or a stroke.
After a stroke, when the blood flow stops and the brain is starved of oxygen, it becomes acidic. And that sets off a deadly chain reaction that destroys brain cells or neurons permanently. But remarkably, the funnel web peptide HI1A stops this process in its tracks, effectively tricking the brain into thinking conditions are normal.
So the channel never turns on, the signal never gets sent to the cell execution machinery, and the neuron survives the stroke.
So without oxygen, the cell just goes kaput, but what you're doing is essentially putting a chop on the message and saying, no, you're fine, stay fine.
Exactly.
It's absolutely fantastic.
It is an exciting molecule.
Yeah.
This extraordinary finding inspired the team to hunt for other tissues in the body that could benefit from HI1A. And that led them from the brain to the heart. Would the HI1A peptide protect this too?
So these are heart cells that we've created from stem cells. So what that means is a stem cell is something that can become anything in the body.
One way to test this is using these lab-grown human heart cells.
Wait, is that the movement of the liquid?
Nope, those are the cells actually contracting.
Wow.
So these are the cells that actually perform the work in your heart. So in order for your heart to pump blood to the body, the heart physically contracts.
That's so cool.
Because these beating heart cells are alive, they can also be killed. That makes them ideal for Meredith Red to use as she tests if HI1A can protect the human heart during an event like a heart attack or transplant.
So during a heart attack or any sort of injury in the heart, there's actually a drop in pH. So we try and mimic that.
As Meredith prepares the experiment, she doses the top row of heart cells, or cardiomyocytes, with HI1A.
It's really nice to be able to have stem cell-derived cardiomyocytes because it gives you an insight as to whether or not the peptide or the pathway that you're looking at is going to be effective in humans.
While the control row below gets none.
So that's the different pHs without the HI1A.
Then she puts them in a chamber that mimics the conditions cardiomyocytes endure when blood flow stops suddenly.
We've got the heart cells in this little sort of incubator thing that sucks all the oxygen out, and this is simulating the lack of oxygen that something like a donor heart might go through when it's in between the donor and the recipient. And we're going to see what this HI1A can really do.
Two hours later, it's time to see if HI1A has protected the cells.
Okay, let's look.
So you can see the darker the color, the more cell death. So this bottom row is our controls.
So this is essentially where there has been no HI1A.
Correct, yes.
But the top?
The top had HI1A.
And
it looks like there's less cell death.
That's pretty.
Or it's lighter in color, so that would mean that there's less cell death in those particular wells.
While HI1A seems effective, it can take up to 15 years for an experimental drug to travel from the lab to your medicine cabinet. And less than 1% of new drugs actually make it to market. However, researchers here are already collaborating with heart specialists to explore how to take the next steps. To see how that HI1A peptide might one day save lives, I've come to see something I've never witnessed before. A heart transplant.
So it's Saturday afternoon and I'm at St Vincent's Hospital in Sydney. And we're waiting for a phone call to say that a donor heart is going to be brought into this room and put into this patient.
This patient has been on the transplant waiting list for 6 months.
He's had viral myocarditis, so his heart's blown out and not working very well. And he's got a little device called an LVAD in there.
He's been kept alive with a mechanical heart pump, and today he'll finally get a donor heart, transplanted by Emily Granger.
The special thing about this donor heart is that it will be actually beating in a box. Obviously, you've got to get the timings right, so you don't really want to take things out and find that the heart in the box isn't any good anymore. Okay, starting? Yep.
As the operation begins, a viable donor heart arrives from interstate with transplant expert Sarah Schuer.
He's got a lot of blood.
Hello, Sarah.
Yes.
I'm Anne.
Nice to meet you.
And is this the heart?
This is the heart. The sound you're hitting is actually the pump beating. The heart's not quite that loud on its own.
When it was introduced 6 years ago, this organ care system doubled the amount of time a heart could be kept alive. But even so, 3 out of 4 hearts never get this far.
So the most vulnerable part is really once that heart has stopped in that donor, we have a pretty narrow time frame to actually get it out, which is about 30 minutes.
So, with its potential to delay cell death, a dose of the HI1A peptide might increase that time frame significantly.
And so we're hoping that by adding in this HI1A, the Venom peptide, to the preservation flush we give the heart, that might extend that time out even up to 60 minutes, which could really significantly increase the number of donors we can use.
And I suppose to give that context though, how many donor hearts are there for people on the wait list?
It's always a shortage.
Right.
So, no matter how many hearts we seem to come up with, there's always more people on the waiting list.
And that's it. That's the heart. The heart should be the size of your fist. So it's a classic heart failure heart.
When it's time to replace the sick heart, Sarah prepares to disconnect the donor heart from the rig.
Pair of scissors.
And that means it'll be without oxygen for more than 10 minutes.
You can see that even when you brought the heart across and handed it over, there's still quite a lot of time before that heart actually starts sort of going again.
It's at this critical moment that HI1A could also offer protection.
Excellent, we're ready to stitch it in. Perfect.
We started looking at some of the small animal models and found that hearts that we preserved with this recovered really, really well. So it's been a real collaborative effort between the Victor Chang Cardiac Research Institute and the University of Queensland, and we've both been doing bits and pieces of the research and bringing it all together. Hopefully something that we can then get into the clinic in the next couple of years and make it even easier for us to do these transplants.
And HI1A's impact on human health might go far beyond transplants.
There's some research that will probably start in the next sort of 3 to 6 months, looking at this in patients that have heart attacks. It's a similar process, damaging it and causing things like heart failure down the track. So we might actually be able to use this even earlier along to try and prevent patients that have heart attacks from having such severe damage. So pretty exciting stuff, really.
Okay, fill the heart.
And all this amazing potential from just one molecule from funnel web venom.
There you go, it's just had a beat.
This operation has been a success.
The patient's new heart is supporting his circulation with just a little bit of support from some medications, but really not much at all.
How exciting.
Yeah, it's great.
I was really moved by what I saw here today.
When the surgeon picked that heart up out of a bowl and put it into a person who needed that heart, it was absolutely incredible. And to think that our little furry spider friends could help us achieve even more of these medical miracles is just mind-blowing.
And saving hearts is just one example of the medicinal possibilities of venom. If there's one thing I've learned on this journey, it's that hidden within our forests, deserts and oceans, there's a vast pharmacy of complex venoms that we're only just beginning to understand. Who knows? Maybe somewhere out here, there's a venom that'll save you.